Anatomy of flowering plants, one of the important chapter in CBSE 11th biology, and some points of this chapter are important for coming Neet exam. First of all, anatomy means study of internal structure of the plant, about internal structure of the plant. So basically in this chapter we are going to study about different plant tissues, and plant tissues are of two types, meristematic and permanent tissue. Let us learn each of this tissue. Meristematic tissue is a tissue that shows continuous cell division. It is of two types. Based on the origin, it is categorized into promeristem present in embryonic stage, primary meristem present in early in life of the plant, and secondary meristem derived from the primary meristem. And based on the location, meristematic tissue is of two types: apical meristem present at the tip of the root and shoot, intercalary meristem responsible for growth in length, and lateral meristem responsible for growth in diameter. Now permanent tissue means these tissue will not divide, the cells of this tissue will not divide. It is of two types, simple and complex permanent tissue. Simple permanent tissue is of two types, three types, parenchyma, collenchyma and sclerenchyma. Whereas complex permanent tissue is of two types, xylem and phloem. And the elements of xylem includes tracheids, vessels, xylem fiber and xylem parenchyma. They are responsible for the conduction of water and minerals along with giving mechanical support. Whereas the elements of phloem include sieve tubes, companion cells, phloem fiber and phloem parenchyma. So they are responsible for conduction of food. In plants we can find three types of tissue system. It includes epidermal, ground and vascular tissue system. Epidermal tissue system consists of epidermal cells, stomata and epidermal appendages. Trichome and hairs are the epidermal appendages. And ground tissue system consists of all the tissues except of epidermal and vascular tissue, that is in ground tissue system. And vascular tissue system consists of xylem and phloem. Then xylem and phloem are of two types, radial and conjoint. In radial type, xylem and phloem are present alternatively, whereas in conjoint it is of two types, conjoint closed and conjoint open. In conjoint closed, xylem and phloem are present together, whereas in conjoint open, between xylem and phloem we can find cambium ring. Then what is absent in gymnosperm? Gymnosperm does not have xylem fiber and xylem vessel, and in phloem it does not have companion cells and sieve tubes. Whereas in monocots, phloem fiber, phloem parenchyma, vascular cambium and secondary growth is absent in monocots. Now secondary growth in dicot stem is of two types, intrastellar and extrastellar. Intrastellar means inside the endodermis, where primary medullary ray develops into interfascicular cambium, and interfascicular cambium gives rise to secondary xylem, secondary phloem and secondary medullary ray. Whereas in extrastellar, the growth is outside the endodermis, where hypodermis develops into cork cambium and cork cambium further develops into cork and secondary cortex. And secondary growth in dicot root includes conjunctive tissue and pericycle, where conjunctive tissue gives rise to secondary phloem, secondary xylem and secondary medullary ray, and pericycle gives rise to cork cambium and primary medullary ray. So balloon shaped structures are called as tyloses. They are the extensions of xylem parenchyma cells into vessels. And protoxylem is present in dicot stem but not in dicot root. And vascular bundles are open because they produce secondary xylem and phloem. And heartwood differ from sapwood in having dead and non-conducting elements in the heartwood, and differentiate between springwood and autumn wood. Spring wood is also called as early wood, and in spring season cambium is very active and produce large number of xylem elements having wider vessel, and these are lighter in color and low in density, whereas in autumn wood or late wood we can find all the properties opposite to spring wood.